sorry for the delay. I'm having a technical problem that they didn't have a good solution for, and the theme of the day for me has been technical problems because I've been trying to deal with my video from last time and having more issues. I usually do these routinely, so it's kind of annoying and slow. It doesn't always happen this way. So I apologize for the delays in getting the videos together. There have been some very odd issues that have come up this term, and I'm not quite sure why or what, but I should have them sorted out quickly. In any event, it's Friday. That means that when we're done here, I'm guessing you have no more classes tonight. Should we all go out to happy hour? Unhappy hour? Okay, I'm going to happy hour afterwards, so I know I'll be happy. So I'm looking forward to the end of class, probably as much as you guys are. So this is good. All right, we've done a fair amount of talking about buffers, and you've seen some practice problems. I went through some examples for you last time, and a couple of you have contacted me and said, hey, can I come in and talk to you? And I'd be more than happy to talk to you. So if you're finding that those are challenging or you're finding that you're having difficulties with those, let me know. Some of the practice problems that I've put on the sheet actually involve a calculator. So don't be surprised if you see that. Those are just for you to practice with. But I can assure you that there's nothing on the exam that you will do that will need a calculator. You can have one, but you're not going to need one. I've also had several people contact me and say, hey, the thing's not on Blackboard. So that tells me they didn't get the message I sent out to them about the fact that, hey, this is not on Blackboard. So if you're not getting messages from me, that probably means you're not checking your own email, and or you're forwarding your own email to another account, both of which are going to be deadly for you. So if you want to stay in touch with messages from me, you need to check your own email and make sure that you're not forwarding it to another account. Because what happens with that is when spam filters kick in at those other accounts, sometimes OSU gets locked out. And you can be completely locked out of messages I get and you never know that they're there. Okay? So just a good piece of advice in general, when you're at OSU taking classes, you want to be using your own account and not forward to another account. That'll cause problems. Okay. Well, we're going to turn our attention now to a consideration of structure. Structure, as I said earlier in the class, is essential for function. We see a relationship. Structure implies function. One of the reasons that biophysicists are so obsessed with structure is because they know that by knowing structure, they can understand how something works. Okay? I have a friend who is a sort of frustrated engineer. He's actually a biophysicist. But when he was an undergraduate, he was very interested in engineering. And so the first thing he does whenever he gets any device, I don't care what it is, it could be the fanciest computer, it could be an iPad, it could be a calculator, it could be a hearing aid, the first thing he does when he gets it is he takes it apart. Okay? He takes it apart because he wants to see how it does what it does. All right? Well, that's what biophysics and that's what structure is all about. We're trying to take it apart. We're trying to understand how it is that this structure allows it to perform the function that it does. So we spent a fair amount of time talking about structure because structure leads us to an understanding of function. The first thing we talk about with respect to structure are proteins. The first things we talk about are proteins. And proteins are obviously critical because proteins, as I mentioned in the first lecture, are the workhorses of the cell. Proteins are going to do most of the work of the cell. DNA and RNA don't really do much work. They store information, they transmit information, okay? But that information is used to make proteins. Proteins are used to make everything else. So if we understand protein structure, we're very well along the way to understanding how it is that a cell accomplishes the things that it does. Most of our interest this term is going to be focused on enzymes. In this lecture and the next lecture, we'll talk a little bit about some other considerations of structure. But most of our consideration of structure will be focused upon understanding enzymes and their structure. All right, so let's start with the building blocks of proteins, the amino acids. Okay? In organic chemistry, you learned something important, I hope, and that is that when a carbon has 4 different things attached to it, there are 2 ways in 3-dimensional space to attach those 4 different things. Excuse me. We can think of it as handedness, if that helps. Okay? They are stereoisomers, another way of describing those. Okay? Stereoisomers. Okay, we talk about an L isomer. We talk about D isomers. All right. Chemists get all wound up talking about R and S with respect to naming things, but fortunately biochemists don't do that. Biochemists by their very nature are lazy. Okay? Biochemists like simplicity because our subject itself is complicated enough. We don't need to give you more nomenclature that makes it more complicated. So we're not going to talk about R and S, for example. Okay? We're going to talk about D form and L form. And no, I'm not going to ask you, for the most part, to understand or show me what a D or an L is. You're not going to have to do that. When we get to talking about sugars later in the term, we'll talk about some very simple considerations for D and L. But I'm not going to say, hey, is this the D isomer, or is this the L isomer, or draw me a D isomer, or draw me an L isomer. That's not what it's about. The important thing with this is to understand that there are two different ways of arranging those atoms in three-dimensional space. Because of that, these guys can have some different properties. Okay? In some biomolecules, because of the arrangement of the stereoisomeric space, that is, the arrangement of atoms in space, you can have one compound that tastes sweet, and you make a stereoisomer of it, and it can taste bitter. Okay? Different chemical properties, and our bodies interact with them in different ways. Okay? So, understanding that these things exist is important. It's relevant for our consideration of amino acids because, with one exception, all amino acids that are used to make proteins have four different things attached to a central carbon. Okay? So this picture on the screen schematically shows a representation of an amino acid, and that representation looks like this. Okay? There's several things on here I'm going to give names to. Okay? First of all, the name alpha is going to pop up in three places. On the left, we see what's called the alpha amino group. It's that group that gives amino acids its name. An alpha amino group. The alpha amino group is attached to the alpha carbon. That's this guy right here. The alpha carbon is the carbon that has four different things attached to it. Okay. I said there were three things with the word alpha in them. The third one is over here. This is the alpha carboxyl group. So as we look from left to right, we've got alpha amino, alpha carbon, alpha carboxyl. Now, when we look at the other two things attached to the alpha carbon, we see there's a hydrogen, and we see there's an R group. You remember R groups from organic chemistry can basically represent anything. 19 of the amino acids have various R groups. One of the amino acids, specifically glycine, has a hydrogen there. Glycine, therefore, is the only exception to the rule that all of the amino acids in proteins have four different things attached to the alpha carbon, because glycine has two hydrogens. It doesn't have four different things attached. That means, therefore, that glycine is, in fact, not a stereoisomer. It's not a stereoisomer. Okay. All the others are stereoisomers. That means that all of the other amino acids can exist in a D form, or they can exist in an L form. Okay? Well, something interesting turns out to be the case. If we look and we examine all of the amino acids in biological systems, they all have a common property. Before I tell you that property, I need to tell you, if I go to it and make an amino acid using a chemical reaction in a test tube, what I will get is what's called a racemic mix. You guys, I think, should remember what that means. And a racemic mix is a mixture. Half D, half L. It's random, and so when you make trillions of these, you're going to get half of them in one configuration and half in the other configuration. Okay? That's if you do it by chemical means. If you do it by biologic means, all the amino acids biologically, with some very, very minor exceptions, all of the amino acids biologically are all in the L configuration. This tells us that there's something stereospecific about the way in which they're made. Something stereospecific about the way in which they are made. It turns out that stereospecific nature in which they're made is that they're all made by enzymes that have a specific stereospecific structure. They will only make one type of amino acid. Okay? This has been conserved all of the way through biology. All right? All of those are in the L configuration. That means that when I am an organism on the face of the earth, I can eat other organisms and use their amino acids, and they will all be in the L configuration, and I can use them to make my proteins, which will also be in the L configuration. Okay? It's very good. It's very useful. There are some very minor exceptions. Okay, I'm going to tell you one of the minor exceptions. I'll probably mention it later in the term. Is there any advantage to making D amino acids? Well, it turns out we see an advantage biologically. Most systems have evolved to use that advantage if they can. Okay? We'll talk later in the term about the fact that there are enzymes called proteases. Proteases break down proteins. That's what they do. They catalyze the breakdown of proteins. You have proteases in your lungs. You have proteases in your digestive system. They break down proteins. You have proteases in your eyes. Okay? Variety of things that have proteases in them to protect against bacteria that might come in and invade. Because those proteases can attack and break down the proteins in those bacteria, killing the bacteria. Well, some bacteria said, maybe if we put something into our proteins that the amino acids, that the proteases couldn't break down, we would be protected. So some bacteria have a few amino acids in their proteins in their coat that are in the D configuration. It gives them some protection against proteases. The proteases won't work on them because they only work on those that have L. Okay? One example. It's a very minor example. Okay? Essentially in this class we will say that all amino acids have the L configuration if they're in a protein. Alright. As I said, there are 20 amino acids that we have in our proteins. Okay? I think you should memorize the structure of all these amino acids. That's a joke. I'm not going to make you memorize all the structures of all the amino acids. Okay? April Fools, right? You're a late April Fool's joke. You don't have to do that. Alright? Now, you will need to know important things about their structure. What are important things about their structure? Okay? Well, one of the important things is that we can group amino acids according to their characteristic side chains, the R groups. They've all got an alpha carbon, they've all got an alpha carboxyl, and they've all got an alpha amino. The only way in which they differ from each other is in the R group. Okay? We categorize them according to what they have in their R groups. I think it's not unreasonable for you to know which category the amino acids fit into. Okay? Which category? I'm not going to make you draw a structure, but you should know that proline is a nonpolar hydrophobic amino acid. Meaning it's got a nonpolar hydrophobic R group. Okay? Now this will help you because when we get down to these last two down here, the acidic and the basic, they have some very important properties that you're also going to need to know. Okay? What are they? Well, acidic means that they have an R group that has an ionizable side chain. Okay? Here is the alpha carbon right here. Here's the alpha amino. Here's the alpha carboxyl. Here's the R group of aspartic acid. Alright? I'm not going to make you memorize the structure, but you should know that aspartic acid has an R group with a carboxyl on it. And you should also know that that carboxyl can ionize. It can lose its proton, just like the alpha carboxyl can lose a proton. Just like the alpha amine can gain a proton. Amines can gain protons. If an amine gains a proton, it has a +1 charge. If an amine loses that proton that it gained, it has a 0 charge. What you see is a 0 right there. When it's NH2, it's 0. When it's NH3, it's plus. When a carboxyl has its proton on, as you see it does here, its charge is 0. When it loses its proton, it has a charge of -1. That's true whether it's the alpha carboxyl or the R group carboxyl. Okay? So, you need to know that. And you're going to get confused about that. So if you didn't get that in organic chemistry, you're going to need to know it here. Okay? Important to understand what happens with ionization of amines and carboxyls. Now, why do we care about that? We care about that because these are not hydrogen bonding things. These are things that are completely losing or gaining a proton. Their charge is changing. Okay? Let's imagine for a moment I've got a protein. We'll talk later about protein structure, but most proteins have a very convoluted, all wrapped up structure. Okay? All convoluted. We call them folded. They actually fold together. They're kind of a cool structure. What can happen is I can have the R group of one amino acid adjacent to the R group of another amino acid in a different place in the protein. Let's imagine I have a carboxyl that's lost its proton over here. It's got a minus charge. And I've got an amine over here that has gained a proton. It has a plus charge. What's going to happen with these two? Well, they're going to be attracted to each other because pluses are going to attract minuses, right? What we will see is the pH that a protein is in, the pH of a solution that a protein is in, can change. That change might, for example, cause the carboxyl to gain a proton. Now the carboxyl, which had a charge of -1, now has a charge of 0. The amine still has a charge of +1. Is a +1 going to be attracted to a 0? No. So are they going to stay as close together as they were when it was plus and minus? No. And so it's going to relax. When we see changes in protein structure, like I've just described to you, we see drastic effects on what proteins do. You're actually going to get tired of me saying over and over and over that small changes in protein structure have enormous differences in protein action. That is what they do and how they do it. So understanding whether the side chains of amino acids are charged or not is really important to understand protein structure. It's one of the reasons we spend time talking about it, and we'll talk more about it as we get moving further along. Okay, let's see. What did I do with there it is. Okay, questions? Okay, everybody's ready for happy hour. So, both of the acidic amino acids are very similar. I showed you aspartic acid, here's glutamic acid. Very similar in structure. All right, there's the alpha carbon. We also see an R group that has a carboxyl. This guy has one more carbon in it than our aspartic acid did, but it has essentially the same simple structure. An R group over here with a carboxyl, an alpha carboxyl, and an alpha amine. Okay. The other group of amino acids whose side chains can ionize are what your book calls the basic ones. I like to think of them as the amine-containing ones. Okay, I want to start with a simple one here, lysine. Okay, lysine looks like this. There's the structure of lysine. Here's your alpha carbon right here. You can always tell the alpha carbon because the alpha carbon will always have an amine and a carboxyl on it. Okay? They'll always have both on it. That'll always be the alpha carbon. In this case, they've shown you the ionized form. The ionized form has a proton on the amine group. It's got a plus charge. Has lost the proton off of the alpha carboxyl. It has a negative charge. And has gained a proton on the R group, which means it has a plus charge. We will see all kinds of possibilities for charges on amino acids. Right? depending upon the pH, all of these groups may change. Because each group has its own characteristic pKa value. You saw last time I showed you some amino acids that had different pKa values. They might have more than one, they might have two. All right, here's one that's got 3, just like the glutamic acid had 3. It had 2 carboxyls and 1 amine. This guy's got 2 amines and 1 carboxyl. Okay? Yes?
Is that what gives that type of amino acid a different isoform?
Is this what gives this type of amino acid a different isoform?
Yeah. Different structures?
No. This won't give the amino acid per se different structures, no. So the different structures are the stereoisomers that I'm talking about arise from the 4 things being bonded right here. Okay? All right. So, this guy, if we added up all the charges on it, we'd say the overall charge here is +1, right? 2 pluses and 1 minus. All right. Now, so you should know the various things that can happen to the amines and to the carboxyl amino acids. All right? If I show you arginine, you'll see a similar thing. Here's arginine. Okay? There's their amine right there in NH2. We see an NH2 as an alpha amine. We see an alpha carboxyl. That means this, therefore, is the alpha carbon. And that's what it looks like. If this guy, we were to put the protons, if we were to ionize this guy, we would say, well, we could put an H there that would make an NH3+. We could put an H there and make an NH3+. We could take an H off of there, and we'd have a COO minus, right? And again, the overall charge, if we did that here, would be +1. Well, do they always ionize that way? No. We might see a proton on, and a proton on. If we had that, what charge would we have? What would be the charge of the molecule if I had that? +2. Remember the carboxyl? See, you've already confused us. The carboxyl, when you put the proton on, it's got a charge of 0. 0 plus, overall charge of +2. What if I took the protons off of everything? What's the charge going to be? No? I hear some numbers. Positives or negatives? What's the charge if I take all the protons off of everything? Okay, charge is going to be -1. Charge of an amine has, with the proton off, is 0. Charge of an amine with the proton off is 0. Charge of a carboxyl with the proton off is -1. Right? So, the overall charge in that case would be -1. You're going to need to do that. Okay? You're going to need to do that. So I would spend some time making sure you understand how those things can ionize, because you will see some problems related to that. Okay, questions about that? Okay. Histidine's a little odd, so I didn't start with histidine, because histidine has its amine within a ring. So you're not going to see an NH2. In fact, you're going to see an NH. This guy will gain a proton, and you see this this N already has 2 different bonds on it. When it gains a proton, it becomes an NH2, but in that case it becomes a +1. This is the one exception where we have an NH2 that's a +1. It's because the N already has 2 bonds. All right, this guy, +1, +1, -1. If we ionize it completely, that'd be an overall charge again of +1. Okay. For all you care about, this is an amine that can gain a charge of +1. That's all it is. You're not going to need to know the structure. You're not going to need to show me that. But you're going to need to know that that R group is an amine. And therefore, because it's an amine, it can be a +1 or a 0. Right? You need to know that. Okay. That's histidine. The other guys, you're going to say, okay, well, it's nice if I know the names and so forth. It's nice if I know the general characteristics of them, but I'm not going to need to know anything about them, because these guys, under the conditions we're going to talk about, essentially aren't going to ionize very much at all. So we're not going to worry about them becoming ions in any way, other than their alpha carboxyl and their alpha amine. Because we always have to think about those, because those can always ionize, but none of the R groups of these guys are going to ionize. There are various abbreviations people give to amino acids, and no, you're not going to need to memorize any abbreviations. Okay? If you want to, and this is also true in the class, anytime you want to use an abbreviation, any abbreviation I use is fair game. You are welcome to use any abbreviation that I use, but the abbreviation must be exact. So if I call something, I don't know, let's say GLU, and you put GUL, that would be wrong. All right? So if you're in doubt, probably better to spell things out. Okay? Well, you should be spelling it pretty closely. Let's put it that way. So spelling is I better be able to figure out what it is pretty carefully. You can't be too far off. Okay? We don't guess for you. Okay. There are some unusual amino acids. And these unusual amino acids are kind of cool. I'm going to tell you a real brief story about these guys here. Okay, these first two. These guys we find in these two on the left. We find this is a thyroid hormone right here. It's called thyroxine. All right, your thyroid gland makes that. All right, so we're not going to worry about that for the moment. We're going to focus on these two over here. These two are related. Oh, by the way, I should show you something else here too. One other thing. All right. So there's another amino acid that's kind of cool. We'll talk about this in a little bit. It's called proline. Okay, proline. If I can find there it is right there. Okay, proline has an unusual R group. Okay, here's the amine. This is the alpha amine, and you can see the R group. There's the alpha carbon. The R group comes around and connects with the alpha amine. It's the only one that does that. The R group connects with the alpha amine, meaning that this alpha amine is not free. It's not free. Okay. This guy can also gain a proton and become NH2+. All right? Because again, the N has two different bonds already. All right? Now the reason I point this one out to you is when we talk about protein structure later, we will discover this guy has some constraints. It can't rotate as much because that ring, that closure of the ring, keeps it from rotating properly. And that has some big effects on protein structure. So that's proline. I show you that because in the next slide here, some of the unusual amino acids, one of them is hydroxyproline. This is the same one I just showed you, but it's got an extra hydroxyl group on it. No, you don't need to draw the structure. Don't worry about that. Here's hydroxylysine. This was one of the negatively charged amino acids. I'm sorry, one of the amine-containing amino acids on the R group, and we see it gained in this case an extra OH group. Well, what's the significance of this? Well, the significance of this is kind of cool. Okay. You have a protein in your body, the most abundant protein in your body. Does anybody know what the most abundant protein in your body is? Bet you wouldn't guess. It's what? I said it didn't hear you. Myoglobin would be a good guess, but no, it's not. The most abundant protein in your body is collagen. Collagen. Collagen is literally the glue that sticks you together. It forms connective tissue. It does all kinds of stuff. It's actually a fibrous protein. We'll talk about fibrous proteins in a little bit. What holds collagen together, collagen has some different coils of amino acids that are arranged in fibrous nature. And they're bundled together to give strength. Okay? So I like to compare it to, let's say, braiding your hair. Okay, my hair is not quite long enough to braid. I used to braid it actually, but your hair is braided. Okay, so she's got her hair braided, right? And that hair braided is actually fairly strong, but that braid will not hold unless she puts a rubber band in it at the bottom, as she has done. If I take and I braid some strands and I want them to hold, I can't put a rubber band on them. I actually link the bonds of them to each other. They chemically bond with each other and make a covalent bond. That holds the strands together. Okay? Well, the bond forms in collagen between amino acids that have hydroxyprolines and hydroxylysines. It's the hydroxyl groups that actually form a covalent bond. That helps the collagen to stay together, because if you don't put those bonds there, the coil comes undone, just like your hair would come undone if you let the rubber band out. Okay? Well, why do I tell you that? It turns out, and for all you nutritionists, this is where you should prick up your ears. It turns out that the thing that helps to catalyze that bond is known as vitamin C. Vitamin C is necessary to make that bond inside of your collagen. Now, you may or may not know that if you don't have vitamin C, sufficient amounts of vitamin C in your diet, which is very unusual in today's world, but if you don't have sufficient amounts of vitamin C in your diet, you will develop a condition called scurvy. And when scurvy happens, those bonds can't form. The collagen is very weak. The glue that holds you together is very weak. And you are very weak. The way they discovered the need for vitamin C was back in the old pirate days. When the pirates, the big, honking, ugly, stinking men who went out on ships, mean old guys, they were always guys, you know, mean old guys, great big burly guys went out on ships, and they were gone on ships for several months, and on those several months they ate salted meat because they didn't have refrigeration. They didn't have any vegetables. They didn't have salads. Okay? They didn't have anything in their diet that was a good source of vitamin C. They went out to sea and they became deficient in vitamin C, and these great big hulking guys that went out came back as little wimpy guys because they developed scurvy. They were falling apart. Right? So if you have a bully that's picking on you, the best thing that you can do, you have to wait a while, but just deprive them of vitamin C, and over time, you'll be able to punch them and do something with them. Okay? All right. So that's, I don't encourage that, by the way, but okay. Okay. So, structure and function. Structure important are important. All right. Let's talk about ionizing, okay, because that's what I've been sort of alluding to here. Now we can see some of the ionization that happens with amino acids, okay? Here's a simple amino acid. It only has two ionizable groups. It has an ionizable alpha carboxyl group, and it has an ionizable alpha amine group. Okay? We start this guy with all the protons on, meaning that this would be at a very low pH. We had a great abundance of protons, and therefore everything that could grab a proton got one. We put all the protons onto this simple amino acid. That means we put a proton onto the carboxyl group, the alpha carboxyl. We put a proton onto the alpha amine, and it would have a structure that looks like this. It would have a charge of 0 here. It would have a charge of +1 here. And the overall charge would be +1. If we take that solution that contains this guy, which is very acidic, and we start adding sodium hydroxide to it, what is going to happen to the protons in the solution? They're going to get gobbled up by the hydroxides. They're going to decrease, right? And they're going to start disappearing. Concentration of protons will start falling. Some of these protons that are on here are going to start coming off. Are they going to come off as fast here as off of here? Turns out the answer is no. And you actually know the answer. I could give you a pop quiz right now, which I'm not going to do, but I could give you a pop quiz right now saying which one is going to lose its proton first. How would we decide? What's that? The carboxylic would be the first one to come off. And the way that we would decide is we would decide by which one had the lowest pKa. Because the lowest pKa is the strongest acid, it's going to be the one that's going to come off first. And it turns out, as she said, this is the carboxyl group. Okay? Well, you saw it right here. In fact, you can see it right here. What's happened is, this guy comes off, and there it is. Let's say I had a pH of 2.34. What am I going to have in that solution at a pH of 2.34? Anybody want to take a chance? Describe it for me. Nobody wants any extra credit. So when I say extra credit, all of a sudden people want to participate, right? Okay. Yes, we would have half and half. Half of the molecules would be in this configuration, and half would be in this configuration, because we would have half salt, lost the proton, half acid, has the proton. Okay? We keep adding sodium hydroxide, pH keeps going up, and we get to a pH of 9.69. What are we going to have? We're going to have half this and half this. This guy's lost both protons, has a charge of -1. This guy has 1 minus 1 plus. This has a charge of 0. When a molecule has a charge of 0, we call it a Zwitter ion. A Zwitter ion. Anybody here know German? Okay. Somebody told me, and I don't know if this is, do you speak German? What does Zwitter mean? And what? So I don't know that. I don't speak German. Somebody told me that Zwitter means hermaphrodite. Yeah. So you've got, it's got both charges. It's got both both groups there, right? Okay. I don't know if that's true or not, but it always made me remember what zwitterion actually means.
Zwitterion is one that has a negative and a positive.
So it is a 0. It could have 6 negatives and 6 positives. It would have a charge of 0.
Neither also just be 0.
As long as it has an overall charge of 0, it's a zwitterion. Okay? All right. So these are the possible charges that would be in this simple amino acid. Pretty cool, right? Let's take a look at another one. Let's say that we've got... Well, actually, this shows the graph. Let's do the graph and then I'll show you another one. Okay? This shows what I just showed you a second ago. All right? What I just showed you a second ago. But now we're going to plot it as a titration plot. Titration plots have pH on the y-axis, and they have number of moles of hydroxide on the x-axis. We start out, as I said, with a lot of protons, which means we start out at a very low pH. We start out down here. That's what we have before we added any OH. As we start adding OH, we see, oh, this guy starts rising. The pH starts rising until it gets to within 1 pH unit of the pKa, and then what happens? It starts to flatten out. Meaning the pH doesn't change very much, it doesn't rise very much as we keep adding a lot of OH. But then we see it start rising again. Why do we see it start rising again? This is Friday, isn't it? You remind me about the guy that got fired from the orange juice factory. Did you know about the guy that got fired from the orange juice factory? I'm sorry?
Because it has a 3H, so it has the ability to lose another proton, so it will change.
So this guy, well, not quite. Let me come back to that. Okay, but you're on the right track. You're on the right track. Okay. You bet the guy that lost the job at the orange juice factory. He couldn't concentrate. Right? Now you know why I thought of you guys, right? Okay. I'm sorry. So, your point over here is that we actually have another proton that can come off, but that's not why the pH goes up. The pH goes up because we have exceeded the buffering capacity. Does everybody remember that? We said buffering capacity was a function of what? Concentration? There's only so much this buffer is going to allow. So once we see it start rising, we know we have exceeded its buffering capacity. It's not buffering over here. That's why the pH is going boing as we add just a very tiny amount of OH. And then all of a sudden it starts leveling out, and when it's leveling out, we are in another buffering range. The buffering range corresponding to the pKa for this. You notice that down here we have half and half at the pKa. We have half and half at this pKa. So this is a titration plot, and this is what it looks like for a simple amino acid. There's another thing on here called pi. Pi is the pH at which a molecule has a charge of exactly 0. Okay? We'll talk about that later. I'm not going to talk about it here, but we'll talk about that later. Okay? All right. Let's look at a more complicated one. Here's histidine. Histidine has an R group that can ionize. Now you notice on that last one I showed you, we saw 3 different molecules, right? 1, 2, 3, right? One that had a charge of +1, one that had a charge of 0, and one that had a charge of -1, right? How many pKa values did we have? We had 2. People always get puzzled, I think, well, we've got 3 molecules, we should have 3 pKa's, but we don't. Right? Because we have half and half, and we have half and half. There's a pKa for each ionization. There's 2 ionizations to get 3 molecules. Look at here. We've got 4 different molecules. How many pKa values do we have? 3. The pKa doesn't refer to the loss of the proton. It refers to the place where it's half lost. Right? Over here, we've got a solution that's full of protons. It's got all the protons anybody wants. All the protons load on to all the groups on this molecule. We see an overall charge of +2 because the carboxyl has a charge of 0, the alpha amine has a charge of +1, and the R group amine has a charge of +1. This game, they've actually rearranged the bond. There it is. Okay? Overall charge of +2. We lose the alpha carboxyl first because the pKa value of that carboxyl is right there. It's for that one. This is the first one that comes off. Notice these guys haven't changed a bit. They still have charges of +1. The next group to come off is in this case, is the alpha amine. It loses its charge over here. It becomes a charge of 0. 0, -1, +1. Here we have a charge of 0. Okay. Finally, we lose the last one. We get a charge of -1. Okay. So that's what this guy looks like. Right? If I did a carboxyl, maybe it'd be good for you guys to practice a carboxyl and see what you come up with if you do the same thing. You'll see it's not exactly the same as this. Try a carboxyl. Carboxyls and amines are going to be different in terms of, that is, a carboxyl, if you have an acidic amino acid. Okay? Something that has an acidic R group, a carboxyl in the R group, try that. Do this exercise and see what you come up with. Good way for you to practice. Okay. All right. Now, I'm very briefly going to introduce peptide bonds to you, and then we're going to move on. By the way, there's a pKa table here. You are not, underline not, going to have to memorize that table. Anything off of this that you need, I will give you on an exam. Okay? You won't necessarily see it in this form, but I'll give you anything you need from this off an exam. Okay. Peptide bonds. Peptide bonds are formed when two amino acids join together. When two amino acids are joined together. To join two amino acids together, I take the alpha carboxyl of one and I link it to the alpha amino of the next one. Alpha carboxyl gets linked to alpha amino. They're trying to show you this right here. Okay? That's kind of gibberish in looking at it, so I'm not going to show you that. All right? I will show you more clearly here. Here's one amino acid. Here's its alpha carboxyl. Here's another amino acid. Here's its alpha amine. The joining of these two guys right here, as you can see here, causes water to be split out, and a bond is left in its place. That bond is known as a peptide bond. So if I ask you on an exam what bonds hold together the amino acids in a protein, I hope that you would tell me that peptide bonds do that. Peptide bonds hold together adjacent bonds in a protein. Okay? Because of this, we can see that every protein has an interesting property. Notice I put these two guys together. Here's an alpha carboxyl, here's an alpha amine. If I had this together, I would call this a dipeptide, meaning two peptides or two amino acids joined together to make what we call a dipeptide. If I had three, I would call it a tripeptide. If I had many, I would call it a polypeptide. Okay? But in each case, every peptide that I make would always have left one free alpha amino and one free alpha carboxyl. The ones in the middle get gobbled up. That's what's happening here. The ones in the middle get gobbled up. And that's going to turn out to be something you're really going to like a lot. Because you don't have to worry about charge of these guys. All the internal alpha aminos and alpha carboxyls don't have charge. When we go to find charge of a protein, which you're going to do at some point, we have to think of three things. What's the charge of the alpha amino? What's the charge of the alpha carboxyl? And what are the charges of the R groups? All the other alpha aminos and alpha carboxyls we won't have to worry about. Very much of a simplification. Okay? I try to keep things as simple for you guys as I can. Okay, it's been a long day. I would propose we finish it with a song. What do you say? Now, what I was trying to get was actually have a very cool recording of this song that someone made for me, and I couldn't bring it up. So we're going to sing it, but it has to do with amino acid. Okay? I will ask you, how did you learn your ABCs? With a song, right? And everybody knows that song, right? A, B, C, D, E, F, G, H, I, J, K, L, M, N, O, P. Lysine, arginine, and histidine. basic ones you should not miss. Allelu, val, il, and met, fill the aliphatic set. Prolene, bens, and cis has S. Glycines, R is the small s. Then there's terp and tyre and phe, structured aromatically. Asp and glue side chains of R, say to protons au revoir. Glutamine, asparagine, bear carboxamide amines. Threonine and tiny ser have hydroxyl groups to share. These 20 amino A's can combine a zillion ways. See you Monday.